small part of the body mass, right? Is only 1% of the body mass. They do get 20% of cardiac output, the volume of blood that actually going through the kidney. So they do have to filter entire plasma. I hope you kind of appreciate the work. So the entire plasma does need to first filter into the nephron, and then the kidney has to deal with needs to go back, what's actually going to be excreted, and it's quite a sophisticated system over there. So, and they do that 60 times a day. So, pretty amazing work. So, which means that's about 160 liter of the filtrates per day. They do use significant amount of energy, and that's for transporters. Millions of little functional units that we call nephrons, which we will discuss in a minute, and a lot of tubules, right? 50 miles worth of tubules where precise, calculation of what goes back into capillary once gets excreted is taking place. And the other very amazing part, as you know, people do survive with one kidney, right? Mm-hmm. If they remove during disorder or they donate that. But also 25% of one kidney could still do a job necessary. Of course, it's a very dangerous should anything happen to that. But all right, and so those are the key functions, right? So we'll talk about the key functions of this. So we're going to talk about the anatomy, and then we're going to still use those terms that you're already familiar with, and we just make sure that they mean. So what exactly those terms mean here in the kidney situation. Excretion is pretty straight up, right? But we'll go through all these terms in a minute. Okay, let's look at the kidneys. So first of all, as hopefully everybody understands right away, is the regulation of the extracellular fluid, right? So, and because we are dealing with the fluid regulation, then the blood pressure, right? Is going to be a big factor. If you have a very high blood pressure, what do you think is going to happen to your urination rate? Miranda?
Oh, is there things will go up?
Yes, definitely. Regulation of osmolarity is another very, very important portion because remember, the kidney is going to be filtrating and dealing with a lot of the electrolytes, so they could put more or less sodium, potassium back into circulation. They are the secondary, very important organ for the pH regulation, lung being the first. With hypoventilation, that would be the fastest way to control the pH. I mean, I am talking about very kind of quick regulation, but the kidneys is going to have very important role because they could increase or decrease expression of the hydrogen ions, and that would be their contribution to the pH balance, and of course the waste, and also they produce some hormones, and the one hormone we already know about. Which one did we already mention and say it's made in a kidney? Erythropoietin. Okay, so they are retroperitoneally, meaning they're actually outside of abdominal cavity. Most of you have taken anatomy, so here is our kidney, which is of course two organs. The renal artery brings the blood into the kidneys, and renal vein takes the venous blood away, and then we're not really going to be focusing on adrenal gland today. Okay, so here's our renal artery, and of course the renal vein. So this part that's light is called renal cortex, and the one that's kind of pink here is renal medulla. Okay, so medulla, cortex. So now let's look at the functional unit of the kidneys called nephron. So the best way to probably kind of start, perhaps, is start with the afferent arteriole, and it will bring blood. It will kind of change it to capillary. It's kind of the like a network of capillaries, and they will bring blood into what we call the Bowman's capsule. There will be a better figures later on. And then from there, the blood flows into afferent arterioles, and then into peritubular capillary, which you guys all correctly notice. And then the blood collects in what we call vasa recta, and then will join the venous return. So the part of the cortex that usually contains the nephrons and their tubules, but many tubules actually dip much deeper into the medulla. So both of these are considered to be nephrons, but this one also has an additional name. They call it juxtamedullary nephrons, that one kind of deep into medulla. And these are the collective tubes that will actually carry the excretion, the formed urine. So on a IHC, this is how the glomerulus look like. So it's kind of network of capillary, and this is the Bowman space. So these guys all, hopefully you can recognize them. And the tubules, they have a bit more kind of sort of, it's a smaller diameter. So all of these, the rest of it really is the tubules. And as a nephron is over here. The term glomeruli is actually refers to the network of the capillaries, and the nephron is the entire structure. Okay, here we are, zooming in. So please note, on this particular picture, the afferent arteriole for some reason kind of put in a different direction than the one before, but it kind of doesn't really matter for what we're talking about right now. So the afferent arteriole brings the blood in for filtration. So this particular area is very, very important because the filtration will depend on a few different things. Of course, the condition of the capillaries and how leaky they are, but then there is actually a pressure that comes from the fluid that is inside of the Bowman capsule. So the Bowman capsule pressure will be at the opposite force of a filtration. Also, the podocytes, the cells that kind of sit on the top of the capillaries, depending how developed the podocyte network, it will also play a role in the filtration, because besides just going through the endothelial cells of the capillary, the blood will also have to filtrate through the podocytes. So this is all going to kind of play a very important role. So the Bowman capsule is just like a layer of the endothelial cells over here, and this is what we call Bowman space, or capsular space. And right here we will start with what we call proximal tube. Proximal meaning in the close proximity to the Bowman capsule. But we'll discuss them when we see all of them. So on the histology, this is what that is going to look like. This is the Bowman space, Bowman capsule, capillary with the blood. It'll be kind of hard to see the podocytes on this one, but they're kind of a little different than the capillary cells. So this is sort of like that particular area. So so what we're going to be talking about today is how exactly the blood is filtrated, how the reabsorption takes place. So reabsorption is putting the part of a filtrate back into cardiovascular system, right? The reabsorption. Secretion will be from the blood to the lumen, and the lumen is the space, of course, in the tubules, right? And then excretion would be removing the filtrate from the body. And let's talk about all these forces that's going to be responsible for filtration rate. the capillary pressure, the hydrostatic pressure of the blood, and then the colloid osmotic pressure will all play a role here as well. So I guess the best way for us to talk about it, let's see. Yeah, we're gonna talk about just in a couple slides. Let's just, because it's easy a little bit. When we have the better presentation here. Okay, let's see. So, this is kind of an interesting statistic, right? So that even though the entire blood plasma is actually passes through the capillaries, only 20% of the plasma is filtered. And it's only 1% of filtered fluid is actually excreted. So here's the terminology one more time. Make sure that you guys really remember them well. Filtration from the blood to the lumen, reabsorption from the lumen into the blood, secretion from the blood to the lumen, and excretion out of the body. So what do we call when we selectively remove molecules from the blood and add them to filtrate? Ben? Brian?
Filtration?
Wait.
Sorry, what was my bad? You said from the.
Hold on. A process that selectively removes molecules from the blood and adds them to the filtrate. The word selective is very important here. What do you think? Tyra? Secretion. All right. Now let's look at this whole setup. This is a good place to start. Okay, filtration would be in this purple color, reabsorption in green, secretion in yellow, and excretion is not yellow, blue, and the excretion and in yellow. Okay, so glomerulus, right? The network of capillaries. The afferent arteriole brings the blood into these capillaries, and it's filtered. Some of it. What percent of it gets filtered at one time?
20%.
About 20%, right? So the rest of it continues their merry way. So let's see what happens to filtrate. So it enters first the tubule that we call proximal. So it's in the proximity to the Bowman's capsule, right? Here's our Bowman's capsule, Bowman's space. Then we have this very fancy little loop that we call loop of Henle. It's gonna have very important role in the filtration. We'll talk about exactly what happens. And the descending loop of Henle and ascending loop of Henle have a very different roles in filtration. So ascending, descending. Then we have a distal tubule, and then finally collecting duct that is going to excrete to the bladder and external environment. So please know all this terminology, the anatomical parts, and kind of the flow. Now this picture is not how in reality nephron is made up. They kind of opened that up for simplicity. But what we'll learn later that ascending loop of Henle is actually goes up right here in this area. They only just opened that up to kind of show the flow, the direction of the flow, but anatomically, ascending loop of Henle is actually flows by glomerulus. And that's gonna become important a little bit in a little while. So 20% of volume at that one given time gets filtered, 80% continuous. And look at that, over 19% of fluid is actually gets reabsorbed. So when we talked about how much solute is actually excreted, right, into outside of the body. We take into consideration how much volume is filtered, how much volume gets reabsorbed, how much volume goes back, and then we're gonna get the solute excreted, right? So filtration minus reabsorption plus secretion equals excretion. Is that clear? Is it? Yes? Okay. This is another very important slide because it talks about the forces that act upon glomerulus, right? In order for the filtration to take place. Okay, let's take a look at that. So the pH, what is it here? The pH is the hydrostatic pressure. You're already familiar with that when we talked about capillaries, right? It's a pressure that exerted by blood into capillaries. So just because capillary, guys, remember, it's only a thin layer of the endothelial cells. So the blood will filter through. So that pressure normally is around 55 millimeters of mercury. Colloid pressure, which is what? Who remembers what the colloid osmotic pressure is? Haley? The draw made possible by proteins that's in the blood, right? Inviting the blood actually to stay back into circulation due to the protein in plasma is about 30. So 55 pushing the filtrate forward, 30 is asking to filtrate to go back, right? And then there is a fluid, right? The Bowman capsule has a fluid, and the fluid would also exert pressure to the blood that's trying to filter, saying, well, we kind of already have a fluid. So that fluid created by the fluid is also would oppose the filtration. Does that make sense? So hydrostatic pressure favors filtration. Colloid pressure favors return that to the circulation, or I should say against the filtration would be better. And then the pressure of the fluid in the Bowman space also against the filtration, the force against the filtration. So then when we have 55 and minus 45, right, the net filtration pressure is about 10 millimeter of mercury. Is that clear? Is that clear? So more, the hydrostatic pressure still wins, kind of, even though the opposing two pressures amount to about 45. So because of that, there is actually still a filtration that takes place, right? Should this be a higher number, there will be no filtration. So the glomerular filtration rate, relatively constant under normal condition, and as we already said, it's influenced by first net filtration pressure, and net filtration pressure will be hydrostatic minus colloid minus fluid pressure in the Bowman's capsule, right? And also what we call filtration coefficient is like how much surface area do we have available for filtration, how permeable the capillaries are, and not only capillaries but also the podocytes and mesangial cells, everything that makes up the glomerulus, how permeable they are for filtration. Because that can, I hope you understand, that can also change, right? If the capillary is less permeable, then less filtration will go through. So, now let's talk about the setup of the capillaries. Let's see. Yeah, here goes. Something's missing here. Okay, this is that same picture that we looked at. Look, so this is the capillaries, right? Then we have the podocytes. By the way, here on the electron microscopy, you see the processes of the podocytes, like additional kind of wrapping around the capillaries. This is the capillaries, and these guys are the podocytes. So this is electron microscopy here. So what are the component of the filtration here? This is, we're talking about what's happening exactly inside of the Bowman's capsule. So first of all, the capillaries do have holes, they fenestrated capillaries, you guys recall that. The other thing that's super important is that the capillaries have a negative charge on their surface. And what that does, it actually repels the protein from filtering through, because proteins are usually positively charged, and so they, they are, wait, what? Yes, no, they're negatively charged, I was gonna say anions, of course, negatively charged. So proteins mostly negatively charged because most of the amino acids that, there's several amino acids that usually part of the most protein that are negatively charged. That's why, if you guys recall, in the ICF, we were saying like anions, mostly made up by the proteins. So if the capillaries surface negatively charged and the proteins are negatively charged, they will repel, right? So that's additional support, is not to have proteins filter through. Because normally protein should not be found in the urine, right? Unless there is a kidney disease. So, and so then we have these two scenarios. Like, in addition, there is actually, it's very hard to show that. There is very thin layer of the basement membrane. It's like a cellular matrix that also kind of presents like an additional a layer of filtration. It also negatively charged. And then of course the podocytes as the final layer, they create a lot of processes and they envelope the capillaries, and that's also something that filtrate has to go through. So between negatively charged endothelial cells, negatively charged the basal membrane, and the podocytes with their processes, we have the filtration barrier inside of the Bowman capsule. So what that does is actually protects most of the cells of the blood and the proteins to leave. So it's the plasma that should be only filtrated. This is just another representation of what we talked about, but with this we're showing this little skinny layer of the basal membrane that's negatively charged. Here is endothelial cells that negatively charged. This is the capillary. This is the fenestration pores in the endothelium, and then the podocytes. So these are the oldest layers, right, that we're gonna have to go through to end up in the Bowman's capsule space.
When I always hear symptoms of a variety of conditions of blood in urine, different things, whatever, you just probably once heard that. Is that damage to the kidney, or is that like, you think that's something that's not getting filtered correctly? Because you just mentioned how it's only
Usually the blood in kidney could happen due to kidney infection, a damage to the kidney of different damage to the glomeruli themselves. Also, in the females, it could be just contamination of the test during the period. But usually, if we exclude that contamination, usually it's either kidney infection or actually kidney failure. Sometimes very, intense exercise could also lead to the breakage of the barrier. And that's usually has to do with the much higher blood pressure during exercise, and the filtration barrier could kind of let the cells go through. But that should not be, it should be kind of more related to the exercise. So the more severe, typically it would be actually a damage to the glomeruli. And what happens a lot, especially with the people with the chronic and unregulated high blood pressure, is the high blood pressure, the glomeruli, this little network of capillary, they're quite, what's the word I'm looking, a gentle, fragile setup. So a consistently high blood pressure will start actually destroying the glomeruli and could lead to actually kidney failure. And the blood will be there as well, but also a protein. And because if you think about it, it's a very, I like the word I use, very fragile setup. We have only very few layers to get through. So if you continue to increase hydrostatic pressure, this actually starts damaging the capillaries. All right. glomerular filtration rate is in the, so the kidney has a local control of the glomerular filtration rate. Meaning like how much blood can actually be filtered through, because it's very important not to go over or not to go under. So couple of things that you guys need to remember. The first way that GFR, what we call glomerular filtration, is regulated is called myogenic response. So if you have increased blood pressure, it will put a stretch, right, on the arterioles and then the capillary. And locally, the smooth muscles in the glomeruli will contract, and so the blood flow will be diminished locally, and that will decrease filtration rate. But that adaptation has the limit. So it's a small increase of blood pressure will the contraction of the vasculature in the glomeruli could handle that and decrease filtration rate. But if it's a consistent, as I said, and pretty high, unregulated high blood pressure, that myogenic response will not really work. And the next one, a bit more complicated. And so I would advise you to spend a little time here and in the book about the tubuloglomerular feedback, because people get very confused about that. So here's what happens. Loop of Henle, ascending loop of Henle, is actually flows by the glomeruli. And they have a very special modified epithelium called macula densa. We'll look at the pictures and we'll repeat that. So right now, just kind of definition. So macula densa is modified epithelium of the ascending loop of Henle. And then the afferent arteriole that brings blood into the glomeruli, right? Also have a very special cells we call granular cells. Some people call juxtamedullary cells. They secrete renin and different paracrine molecules. Paracrine molecules would work locally. So this is what happens. When you have a higher GFR, so they actually sensitive for the sodium delivery. So macula densa, this modified cells, they could sense higher sodium chloride passage through the macula densa, and then they send the signals to the afferent arterioles to constrict and decrease GFR. So here's this setup. Remember when we were just going through the proximal loop of Henle, it was all kind of unfolded, but in reality, the loop of Henle descending, ascending. See how it flows right between the afferent arteriole? So now if you look at over here, this is the section of ascending loop of Henle. So this is this area, ascending loop of Henle. And see how they have this modified epithelial cells that called macula densa. They sensitive to the high sodium. And here in the afferent arteriole, there is also cells, modified cells, they call granular cells. So it is communication between macula densa and the granule cells that will also result in constriction. And if you have the constriction here, then the less blood enters glomeruli. So this one is called tubular glomerular feedback. So I have them here together, just kind of so that mostly to repeat really. This is everything that we talked about. We'll deal with the renin in a bit, because this is an additional hormones that these glomerular cells can release. But it has nothing to do with this local control. It actually has the body-wide effect. We'll talk about renin soon. So please know that the local GFR regulation consists of myogenic response, high blood pressure, stretch, constriction. And then less decreased GFR to reduce the high blood pressure of this fragile structure, or macula densa cells sending the feedback to the granule cells results also in constriction, vasoconstriction of afferent 1 and decreased GFR. And again, protection of glomerulus against the high blood pressure. Just follow these two slides and also the writing in the book a bit little extra. Make sure that you know the difference between these two. Yes.
The two responses that you're talking about here only affect blood pressure within the glomerulus, not body blood pressure.
Yes, it's a local control to protect the glomerulus from the high blood pressure. Again, to the point, if the high pressure is chronic and very high, that's just not gonna be enough and the damage will be there. But if it's a slight increase of blood pressure, these two will protect the glomerulus from the high blood pressure.
Is it detecting high blood pressure or that sodium chloride?
Two different things. The myogenic response is detecting high blood pressure,
Oh, okay.
and the tubular glomerular feedback is only looking for the HCl flow increase, because you have high blood pressure, you just have a faster rate of the HCl flow. That's what they're sensitive to. For the myogenic response, it's a very simple stretch receptor, mechanoreceptors. So increased stretch, constriction.
Why sodium chloride? What's the issue? I mean, you get more concentrated.
It's only because, like, if you have a high blood pressure, you actually have more flow.
Okay.
That's all. It's increased flow, so they're sensitive to the sodium chloride. This is kind of really what we just talked about it, but kind of, one more time. I think maybe you could just go through this, guys, because it's really the same thing we just said, but it's also kind of step by step, so it's kind of nice. Okay. So now let's talk about how would that look like. So if you do have the high blood pressure, right, so then when we have the vasoconstriction here, we will decrease the GFR. So decreased GFR, vasoconstriction. Increased blood pressure would actually vasodilate. Well, technically, it's really not that much of a dilation, because I have to say that the low blood pressure has much less control over the afferent arteriole. It's more like a normal, slightly dilated. It's mostly the high blood pressure that leads to constriction. And this is the slide that tells you that autoregulation is only to the point, right? So somewhere between, so this is a mean one, right? This is a mean arterial pressure. So somewhere between 80 and 180, you may still be able to be efficient. But after that, it will not be able to actually try to, the blood pressure would not be able to be regulated on the local level, and it will affect the glomerulus eventually. So it's a kind of where the GFR autoregulation local control still works within those limits of the mean blood pressure. So, as we mentioned, the word reabsorption is what? From where to where? Reabsorption.
Blood.
From the
Blood.
filtrate
Filtrate.
to the blood. So many different ways to reabsorb. Sodium usually reabsorbed by the active transport. However, a lot of different chemicals could just go by the gradient. Sometimes they go paracellular. That's how we will depict the paracellular or transcellular. Water usually moves via osmosis. Again, just following the solute. there are multiple transporters that you don't really have to memorize, other than just to kind of understand that the reabsorption is the process by which, it really depends on the kind of solute that is being transported. Some of them go paracellular way, some of them use dedicated transporters. You're not really required to memorize the name of transporter, that's why I'm not giving it to you. The only one that we wanna talk a little bit more is the glue transporter that is dedicated for the glucose, right? That's an interesting one, right? Because this transporter is, so this is the lumen, by the way, this is the lumen. This is where the filtrate is. This is the tubular of the cell, the endothelial cells of the tubules. And this is the interstitial fluid. And this is how it's presented in your book, but please understand that once it gets to interstitial fluid, it's gonna go to capillaries. They're just not depicted here. So from the lumen, through the cells, interstitial and the capillaries. So it's gonna go to the blood. So what happens with the glucose, what should have happened with the glucose, the CGLT transporters, they actually move the glucose and the sodium. The sodium goes down its electrochemical gradient, and glucose kind of rides with it because it has a binding site for both, for the glucose and for the sodium. So even though glucose is usually at the greater, there's more glucose inside of a cell, so that would be kind of pulling the glucose against its concentration gradient. But because the sodium drive is going down its concentration gradient, it pulls glucose with that. So this is the transporter that could get oversaturated. Theoretically, if you have too much glucose, it just would not be able to work. So some glucose will escape it to filtrate. And then on the other side, we call it basal side, we call it apical side. The apical side is the one that closest to the filtrate, and then the basal side or basolateral, sometimes pupil side, is the one that's on the opposite one, close to interstitial fluid. We have a dedicated transporter, the glue transporter, specifically for the glucose. There are multiple glue transporters, 2, 5, 4. You guys really don't have to worry about it too much. And the glucose will then exit this way and then into circulation. And the sodium, if there's too much, will be actually taken care of by sodium potassium pump that you already know about. So glucose rides with the sodium on a symporter. So we talked about filtration forces, and we talked about how net driving filtration is 10 millimeter of mercury, 55 minus 30 for osmotic, and minus 15 for the capsule pressure, right? But so let's look at this situation here now. The pressure of the fluid pressure in the capillaries favors reabsorption, is only 10 millimeter of mercury. The colloid osmotic pressure is 30. So then you can see that the pressure gradient favors reabsorption because it actually 20 millimeter of mercury. So peritubular capillary osmotic pressure draws the fluid towards the capillary because osmotic pressure. See, do you guys recall this? Our 10 millimeters was what the filtration pressure was in the capillaries. In the tubules, right? Mm-hmm. So following that, if the pressure in the capillary is only 10, right? So then you have a hydrostatic pressure 10, osmotic 20, 30. So the net is 20, favoring reabsorption, favoring going back to the circulation. the reason I'm showing you this particular slide is actually, not to have you memorize all these different transporter, but to sort of, to have you to appreciate, you know, how much going on here, how many transporters are used, and also very importantly, the use of ATP. Because remember I was mentioning to you that the kidneys do use a lot of the ATP. They use a lot of direct transport, secondary, tertiary transport, and what that means is that when you have a direct transport, right? So for example, a direct transfer will be like potassium in, sodium out, right? When you talk about, for example, secondary indirect transport, meaning it's like one molecule is actually moving in and the second one is kind of riding on that gradient. And there is also something called even tertiary inactive transport, where you have, because of the one transporter bringing something in, another one has to remove it on the other side, so you have like a multiple layers of the transport. You really guys don't have to memorize any of this, but to appreciate why so much energy used, why there is so much transporters are used here, and that's because of the secretion, that something that's coming from the blood back into tubules is a kind of very sophisticated machinery, because by now, whatever, majority of stuff's got reabsorbed. So the only thing that's gonna go back to secretion is gonna be very tightly regulated. And so they are very specialized transporters, because you, at this point, you really kinda fine tuning what exactly could leave, because once it's secreted, it will be excreted. Because excretion is equal filtration minus reabsorption plus secretion. There is also something called clearance, and the clearance is very important, especially when we develop pharmaceuticals, is the rate at which solute disappears from the body, right? Excreted, or by metabolism. For example, look at this one. So here we have a glucose candle. In red, you have the glucose, so you have a plasma glucose, that's a plasma. And here you have the filtration, reabsorption, and excretion rate of glucose. So look at the red one first. So this is filtration. What that actually suggests to you is that all glucose is filtered. So it goes into filtration, but it shouldn't go into excretion, right? So how's that happen? Well, this is where our reabsorption takes place. So look at this, reabsorption. As glucose being filtrated, now you look at the green line, it's being reabsorbed back into circulation. But at 300 milligram per minute, when you actually have a very high concentration of glucose, this is where your reabsorption cannot keep up. And glucose will start escaping in the filtrate. Does that make sense? So first, whatever's filtrated gets picked up and reabsorbed. Once you reach this level of the glucose, then all the transporters are saturated, cannot really, because normally if they would not get saturated, the green line would follow the red line. As much glucose comes in, as much glucose goes reabsorbed. But there is a limit to saturation to transporters, right? And so it just starts escaping into the urine. And so as you can see now, right about this, well, they put here the 300, but so this is when the glucose starts showing up in the excretion, because now you cannot actually reabsorb that. Micturition is very simple reflex. It's actually the process of urination itself. So as you know, the urethra bladder and ureter, bladder, and urethra is the way to the excretion. And we do have the internal sphincters, which is smooth muscle, and external sphincter, which is the skeletal muscles. And we have a very simple the reflex from the spinal cord. It does not really involve, you could override, of course, you could override and inhibit micturition, but if your central nervous system is not doing that, at the spinal cord level, the motor neuron will fire to the external sphincter, which is the skeletal muscles, and they will stay contracted. And so in order to release urine, right, you actually have to increase the parasympathetic innervation, and then smooth muscle will contract, which is in the bladder itself. And the internal sphincter will allow, just because of the contraction, it will be pushed open, and the external sphincter relaxes and allows to release urine. I do want to go a little bit into the 20 with the hopes that we'll leave a little bit of time on Tuesday to go through some questions.
Did you finish the last chapter?
Yeah, I don't think we finished
You
18.
didn't finish 18.
I didn't finish 18?
No,
It's like a few
Time
slides
for those
left.
last few slides, yeah.
Where am I? Oh,
We
I'm
stopped
one
on
thing. Hold a
29.
second. And now you're telling me that? After
Well, you were on a roll. Really buzz.
Got obliterated my question today.
I thought we finished. Why wouldn't we finish?
No, we stopped on 29 because you said we were
Oh,
like
did I
looking
feel like,
sleepy.
oh, maybe, I could recall now. I was telling, oh yeah, I recall now. I was telling you, okay, I'm losing you. I do remember that.
Yeah, it really was.
It was very quiet.
You were like all
and people were like, okay, hey, where is it?
I'm like fired up and ready to go today, let's do this.
Okay, got it. Thank you for reminding me. All right, where were we? Oh, yeah. We were.
We're on 29.
Yeah, we left off on 29. So I just,
Oh, really?
Yeah.
Oh my goodness.
you said that it was like kind of a lot to talk
Well,
about.
no, the one before
No, I
that.
do remember Thursday was kinda, I felt like.
No, it's like.
It's
two
the one
more.
before
People
that.
done.
Professor.
Okay,
Professor,
so-
it's like 2 or 3 slides before this.
Oh, 2 or 3 slides before that?
Yeah, it's the one where you, no,
The
2
guy with the
slides, yeah.
picture.
Yeah, I kinda do remember that. I don't think we talked about, we definitely talked about Venus
Yeah.
and then we talked
Yeah.
about that.
This
This one,
one.
I think this is the one, right?
Yeah.
Okay.
Yeah, slide 27 I wrote on exams.
Okay, all right. Okay. So let's talk about this. This is actually talks about how exactly our respiratory control center, amygdala and pons, controlling the ventilation rate, like the control of ventilation, right? So CO2 is the key chemoreceptor. And what's cool about the CO2 receptors, not only we have them into carotid arteries, most importantly, the medulla has the CO2 receptors as well. And then O2 receptor and pH receptor also play a very important role in regulating ventilation rate. So the carotid, aortic, there's also other locations where we have the O2 and pH receptors, but they're the main ones. So this is gonna be the input in medulla, right? But besides this is the plasma concentration, hydrogen ions, O2, CO2. Emotions and our voluntary control, meaning like if you wanna slow down your breath, if you wanna take a deep breath, obviously you can do it, right? And of course, fight and flight response, this is what that branch is. And the limbic system is our kind of main emotional control system in the brain. So this is gonna be the inputs into medulla oblongata to control the respiratory rate. So high CO2, as we already talked during the lab, will activate inspiration and expiration, meaning like it will increase the ventilation rate. And the somatic neurons, if you guys recall, as the skeletal muscles, they will in turn activate the muscles that play a role in ventilation. So, high CO2, increased ventilation rate, low CO2, decreased ventilation rate, low O2, increased ventilation rate, a pH, depending. So in acidosis, what do you guys think is gonna happen to ventilation rate? Acidosis.
That's high CO2 for 7, right?
Increase or decrease expiration rate?
High CO2 increase.
increased respiration rate. Alkalosis, decreased respiration rate. Yes. So peripheral chemoreceptor located in the carotid aortic artery. So all of these are the inputs, afferent input. And then there's the spinal fluid actually also has its own, and that's where the medulla gets the information from the spinal fluid. So this is the example of the blood vessels with the low pH. So we have these specialized cells in the carotid arteries where they call global cells. They have the receptors for the low pH. And so there is a pathway that's going to lead to the release of the neurotransmitters and the action potential to the medulla oblongata. So this is the afferent pathway. And then another part that's very important is that medulla oblongata itself in the brain was also have a receptors for the CO2, and we call them central receptor. And with this one's on the carotid and the aorta, we call it peripheral receptors. And the CO2 is being the main modulator of the respiratory pathway. That's why kind of explanatory how we actually have that in the medulla itself. So, and this would be the direct effect from the medulla to the ventilation rate. So, and this speaks to what we kind of already know, that high plasma CO2 will increase ventilation, and once it's normalized, it will give a negative feedback to the medulla.
This is oxygen to the brain.
Huh?
This is oxygen to the brain.
No, this is a CO2 receptor.
Got it.
CO2 receptor.